analysis platform with integrated databases. Okay, so before I start, I have to admit I'm a pure bioinformatician. I majored computer science as an undergraduate, did my PhD in Penn State, and then started from 2011. I worked with Peter Kwong in NIH and Larry Shapiro at Columbia on the VRC01, neutralizing antibodies. And we got some success. But I think, hey, antibody repertoire sequencing, that's easy, right? And then in 2013, I came back to China and started my own lab. Okay, and then I did some repertoire study. For example, so this is like a clonal distribution of the top 100 clones, antibody clones every chain. Okay, we saw A, B, C, two very highly expanded clones. Then the next question I ask myself, what are they? Okay, what are they? And later on, we collected more than 2,000 Rep-seq data sets. So that's like more than 2 billion reads and 300 million clones. And I found only a minimal amount of antibodies are very well annotated and functionally studied. But I'm not an immunologist, so I want to know, what are they? What are the vast majority of those antibodies are? Then I remembered, well, CDR3, yeah. So that by large define the antibody binding. And then I went to the internet looking for the answers. Remember, when we start a gene, when we want to know the function of a gene, we go to NCBI. We do BLAST, we do search, and then we know the answer. But when it comes to antibodies, there are a lot of databases. Of course, good for HIV-1 researchers because we have neighbors and also the HIV molecular immunology databases. And for the therapeutic antibodies, we have AIRR-seq. But for each one of the antibodies, we can do BLAST, we can search, and the chance of finding exactly the same or exactly the same CDR3s are very rare. So how about millions of clones? That's my question. I'm so confused. Okay, then to help myself out, to help myself out, I want to know how many antibodies do we know? Okay, then we want to collect all the known antibodies and therapeutic antibodies. So we went through all the database information about, and then for these antibodies, we found, in WHO, there are 521 therapeutic antibodies recognized. Okay, and for the other antibodies, actually there are around 90,000 antibodies with known antigen or disease associated. Okay, so what we did is we take these antibody sequences, we annotated them, we aligned them, we assigned VDJ, we extracted the CDR3s and both in amino acid and nucleotides and build a database so that using CDR3 amino acid or nucleotide, we can annotate, as many as possible. And then because of the antibody repertoire is very diverse, so the current studies normally take a dozen samples and then compare about the disease sample against the control. But we think that's not enough. So we collected more than 700 donors, more than 2,000 samples, more than 2 billion antibody sequences. All of them are heavy chains. And we got more than 5 million public clones. Okay, so this 2,500 Rep-seq data set contains different genders across different tissues amplification strategy, immune status, and age. Then what we did later is we use a uniform analysis pipeline to perform the low-level analysis and high-level analysis. Okay, so we do the VDJ assignment, we do clone type analysis, and then we can compare across, whatever you want to compare. And then within this data set, we got public clones and marked them, annotated them. So after we got all known antibodies and more than 2,000 Rep-seq data, we integrated them into a platform we call that Rapid. So what you can do in Rapid, so if you have a Rep-seq data set, so you can select reference from any of these 2,500 data set, and then you can choose to customize your germline reference. Of course, if you don't want to, we have a default reference. And then after you upload your data set, so you can, after a while, you can visit your analysis result. Of course, if you choose a reference, like a set of Rep-seq data, so the comparison result will also be shown. As we mentioned before, so because we collected all the known antibodies and therapeutic antibodies, so for each of the Rep-seq data, we can annotate the CDR3s. So whenever you have a CDR3 with known antibody, with antibodies of known antigen or disease associated, so we can annotate them. For example, we take Roskin's data SRR, this one, and then we perform the Rep-seq data analysis, and we got 20 antibodies, overlap from the known antibody and therapeutic antibodies. Then we get this pie chart to tell you what are those 20 antibodies are. Okay, actually, look at here. So because this data set is an HIV-1 infected sample, so we got 2 HIV-related antibodies, and then we have, this hyperlink. So if you click this hyperlink, it can tell you what antibodies is it. Okay. Then, so this is the functions of Rapid. So on the left side, we have antibody annotation because we have known antibodies and therapeutic antibodies. So basically, we take the CDR3 amino acid sequence or nucleotide sequence. So whenever we got a Rep-seq data, we'll compare the CDR3s, any one of the CDR3s, over the CDR3s in these 2 data sets. And we can also do enriched analysis for diseases. Okay. And we also have the query functions for antibody and rapporteurs. You can do search with sequence, antigen or disease, or free text search. Okay. So after we finish the Rep-seq analysis, so we can have a comparative result of gene usage, CDR3, junctional diversity, SHM pattern, and clone diversity. So now we have more than 7 billion reads, and we want to do something different. Okay, and starting from January 2020, we have COVID-19. And then we want to know for all these samples, how many COVID-19 neutralizing antibodies are there? Actually, we did found some. I'll show you later. And then when we went on to query those antibodies, therapeutic antibodies, and interestingly, even in the healthy people, we found, 14% of the therapeutic antibodies were found in our data set. For example, so this COVID-19 neutralizing antibody in Roskin's, I think so, yeah. So we found 22 donors and 78 samples. So there are 152 sequences, antibodies. They share exactly the same amino acid CDR3 with this neutralizing antibody. Actually, for all the COVID-19 antibodies we reported so now we found 1,500 neutralizing clones, which constitute to 35,000 unique antibody sequences. Okay. Another example. So we both know PD-1 is a therapeutic clone for cancer, and in our data set, we found 14 donors with 30 unique sequences. They share exactly the same CDR3 with this camera, sorry, can't read that, with this PD-1 antibody. So we also did some other therapeutic antibodies, and we found many, 20% of the 521 therapeutic antibodies can be found in our data set. Moreover, if you look at the diversity identity plot, we can see that all these sequences, they are separated. So if we look at the sequences, we found that they came from the different VDJ recombination, but they share exactly the same CDR3, and properly they all bind to their antigens. And this indicates for all these therapeutic antibodies or virus neutralizing antibodies, it's possible to elucidate these antibodies via vaccination. So later on, actually, we did naive B-cell sequencing, repertoire sequencing, and we found, various therapeutic antibodies can be found in the naive B-cell repertoire. And we performed super deep repertoire sequencing with 4 samples. And for one of the samples, we even found 19 therapeutic antibodies. That's from our naive repertoire. Okay, so that's so much for rapid. To summarize, so we have integrated of known therapeutic antibodies and other known antibodies, and we also integrated high-quality Rep-seq data set. We built a uniform pipeline with various options for reference selection, and we can perform systematic analysis and annotation for Rep-seq data. And that's a good resource for public clone identification and integration. I think, in the near future, we'll acquire more Rep-seq data set for various health centers and diseases, tissues, and also probably we'll integrate the computational structure method to identify therapeutic clones, virus neutralizing clones. Thank you.
So thank you, Dr. Zhang, and we can have 1 or 2 questions if there's any. So maybe I also want to ask that you have so many antibody sequences in the RAPID database. So where are these clinical samples from, and are these data being published? So besides query from RAPID for public sequences, can we access the raw data for further analysis?
Well, that's a good question. Actually, there are around 250 samples are collected from healthy donors by our lab, and all the other data sets are downloaded from the internet. So that means they are all published. Of course, we have more than 5 million public clones, and after, I think after we publish this work, so that's accessible to everybody.
Thank you. Any other questions from our on-site attendees, and also from the online? Okay.
Well, if
Okay.
so, yeah, most of these therapeutic antibodies were natural antibodies, correct? And but you're seeing how common they are in normal, in general populations, right? Is that right?
Yeah.
Yeah, that's neat. Yeah.
Yeah. Actually, you know, for PD-1, for anti-PD-1 antibody, actually we found, you know, exactly the same CDR3 in mice and also in zebrafish even.
That's amazing.
Yeah, you know, I'm not a biologist, but I'm really perplexed why they exist and what they're gonna do. You know, that's probably the next question we are going to follow.
Yeah.
Thank you for your question.
Okay. Any other questions? Okay. So if not, let's thank again for Dr. Zhang's excellent talk.
Thank you.
Okay.
Thanks.